So let's now go through the actual visual eye overall transduction process. When we take a look at our eye, we want to consider our different parts that we have. So first of all, we have the retina, which is in the back of the eye. It's this entire back region of our eye. And this is the area of the eye that is very sensitive to light. So what happens is light comes in through the eye and goes all the way to the back. And it's in that processing in the back of the eye is where the receptors actually are. And the retina is made up of two different kinds of receptors. We have our photoreceptors, which are there for light. And then we also, we have our photoreceptors, and there's two different types of photoreceptors. There's rods and there's cones. So the rods are the types of photoreceptors that are sensitive to light. So this is what tells you whether it's daytime or whether it's nighttime. On the other hand, we have our cones, and our cones are sensitive to colors, but not actually to the light. So this is where we get our reds and our blues, our colors of the rainbow are where our cones are coming from. So as these specialized photoreceptors work together, the rods and the cones, we're able to eventually combine the information about light and about colors. There's a very particular part of the retina that we also want to be aware of, and this is the fovea. The fovea is the area of the sharpest vision in the retina, and it's an area that's highly condensed with our photoreceptors, particularly our cones. There's a very high majority of cones in this fovea area, which allows us to get a lot of detail. Now, as the information in our brain and our eye is processing, we have the light that comes in, it hits the retina in the back, and it goes through the photoreceptors and a few other cells that are in the retina. Once that information is processed, it's going to essentially go from the front of the eye to the back of the retina, and then it bounces forward in the retina. And when it bounces forward in the retina, it then is going to travel down the optic nerve. And the optic nerve is what's actually connecting that eye to the rest of the brain. So because the photoreceptors are where this actual processing is happening, where the transduction is going on, we then take that information, which is now a neural impulse, and we're able to travel it to the rest of our brain. Where this optic nerve actually exits the eye, this is called the blind spot. And the reason that it's blind is that there's no photoreceptors on it, so we're actually not sensing color and we're not sensing light on the part that optic nerve is actually leaving the eye. In terms of the eye overall, we can see some of the major pieces. So we have our cornea and our lens at the very beginning, the light's going through. We notice that in one area, images are up and down. So for example, when I actually look at something, I have a top and bottom relationship. When it goes through your eye and it goes through the lens, it actually gets reversed. So what actually shows up on our retina is a reversed view of all of the images. Now, that does get turned into a neural image. Once it gets into the perception process, that's when we put it back up. That's when we go, okay, things aren't actually upside down. Let's go ahead and take a look at everything the right way through the perception process. But it's good to know that the actual sensation that we end up getting is actually reversed. And here's a closer version of the retina. So again, thinking about our eye, what's happening is that the light is coming in, it's going all the way to the back of the eye, and then it's bouncing forward. So what we're seeing is the very back of the retina first. And this is where those rods and the cones are. So again, the rods have to do with our light perceptions, our cones have to do with our color perception. And as that moves forward, so the light came down, it hit the rods and the cones, and then it bounces upwards. And as it's bouncing upwards, it's also going to hit bipolar cells and also what are called amacrine cells, which I'll show you in just a moment. So our bipolar cells are kind of in this middle level, and they're bringing together the information of our rods and our cones. And then we also continue on up and we hit our ganglion cells. And our ganglion cells are again pulling together this information. Now, the bipolar cells are specific to rods or specific to cones. So, for example, you'll have one bipolar cell that's combining a whole bunch of information from rods, and you'll have another bipolar cell that's combining a whole bunch of information from cones. But then what's happening is those amacrine cells, which here's another picture for you. So again, if we're thinking about this as the light is coming in from the front, it's hitting the back, and then it's bouncing forward. So now we have the back of the retina is actually at the top of this picture here. And as we're going forward, again, we hit our rods and our cones first, which take in that light. We then hit these bipolar cells. And these bipolar cells, which are specific to the rods or specific to the cones, are combining this specific information. But we need to make sure that we're actually connecting all of the light and all of the color information. And that's where those amacrine cells come into play. So the amacrine cells, if we go back to our processing, originally I had labeled them 1, 2, 3. Our rods and cones are still 1. Our bipolar cells are still 2. Our amacrine cells are now really a third layer of cells, and then the ganglion cells move up to a fourth layer of cells that the light is actually going through, and now in this case neural signals are going through. So the amacrine cells are pulling together the bipolar cells into one complete message, and then the ganglion cells take those messages, condense them even further, and then send them directly to the optic nerve, which is really your fifth step. So the transduction process is happening at the rods and the cones. That's where your photoreceptors are happening, because that's the first place that we turn our stimulation into our neural message. However, the ganglion cells are really the closest thing to the optic nerve. So just let's make sure that we connect this back to the brain. I bet you know this answer, right? Which lobe of the brain processes visual information? Well, you're probably going, there's 4 lobes. You might be saying, I know it's the one in the back. And it is the occipital lobe. The occipital lobe's at the very back of our head, the base of our skull essentially, and that's where all of our visual processing is happening. And just to give you that idea, knowing that the occipital lobe is in the back, when we take a look at our images, it's important for us to recognize how our eyes are actually processing. So what happens is both our left eye as well as our right eye see everything in front of us. So in this case, both the left eye and the right eye are seeing both the guy and the girl. And when we take a look at this information, we can say, okay, all right, so they're both bringing in the guy and the girl. We know that they're also reversing the images, so instead of looking at the people standing up, we're actually looking at them upside down. But note how each eye then splits up. So for example, our left eye is going to send some information to the left side of our brain, but our left eye is also going to send some information to the right side of our brain. So in this case, what's happening is the guy is essentially going to the right side, and the girl is essentially going to the left side from our left eye. But let's switch it up. Let's look at our other side, because from our right eye, we also have a split going on. And in our right eye, we have some information going to our right side. In this case, the guy's on the right. So again, the guy is going to our right side. And then part of the right eye goes over to the left brain. So now the girl is ending up on the left as well. So ultimately what's happening is that our eyes are switching up this information and setting it so even though each eye is taking in the full stimulation, only parts of the brain are actually receiving the full imagery. And where this information, where these optic nerves from each eye actually combine, is what's known as the lateral geniculate nucleus, or the LGN. And we have one of these on each side. So we're basically getting a little bit of the left and a little bit of the right, connecting on each side of the lateral geniculate nucleus. From there, the information is then going to go all the way to the back of the brain. We're going to see the full processing. So let's see, we'll switch that over to orange. So now I have all the pictures of the girl are going just to the left side of my brain in the primary visual cortex, and then the meaning is being associated with it. So for example, if I know who she is, that's where our visual association cortex is going to come in and actually provide us meaning. Similarly, all the pictures of the guy are on the right side of my brain and are being processed completely on the right side. So now that we know how the eye actually takes in some outside stimulation and is processed, we just want to make sure that we're clear in terms of what this stimulation means. So the eye itself does not actually take in any objects. That's all of our brain creating the perception once that neural image has come in. So just going back to the stimulation, it's important to recognize that the information that's actually coming into our eye consists of wavelength and amplitude or intensity from the actual light wave itself. So for wavelength, we're talking about how far or how close different waves are to each other. And this is telling us, based on the electromagnetic spectrum, whether we're seeing red or blue or green, for example. Whereas on the other hand, we have our intensity or an amplitude. So how high or how low. That's going to tell us our brightness. So whether it's daytime or whether it's nighttime is going to be coming across with our overall amplitude. And what's important from this is to recognize that not only our actual daytime versus nighttime, but also our color is something that's based completely on an outside stimulation, and it doesn't actually exist. It's how our brain is processing these different kinds of wavelengths. So it's our perception that ultimately allows us to see the color, not the stimulation, not the sensation itself. And with that in mind, as we go through and we look at things like color, we can just take this picture and say, okay, is this person really see-through? Well, when you look at this, you go, well, no, of course, she's not see-through. That doesn't make sense. She's a person. People aren't see-through. This must be some sort of optical illusion. There must be something, maybe it's been photoshopped or something along those lines. Well, really what's happening here is this is what's called an invisibility cloak. And they do exist, and ultimately what's going on is the light is reflecting. So everything that's coming from behind her is actually being reflected on the front, giving the illusion that she's looking like she's see-through. And what this shows us is that even though what we're seeing, so in this case what we would be seeing would be the people behind her that are moving around, our brains still tell us in the perception what's actually happening based on the confines of our understanding of reality. And this is a clear differentiation between the sensation and perception, because here what we see happening and what we believe happening are definitely very different things. Now, with this idea of sensation and perception, we just want to make sure that we're very clear as to whether or not most people sense colors, sounds, textures, odors, or tastes in much the same way. So as you're looking at this question, you're like, okay, most people looking at things the same way. Well, there's different light waves, there's different sound waves, and the answer here is true for the sensations, because ultimately what we're taking in are those outside pieces of information, and we are turning them into neural images. Everybody's neural images will be the same, but then our perceptions and what we actually determine is happening is what's going to be different. And just to wrap this section up in terms of what we're seeing, what we're not seeing, I just want to make you aware of the electromagnetic spectrum. And again, this is showing you the differences in the wavelength and also the color schemes and ultimately what kinds of colors we're seeing. And you'll notice that as humans, in terms of our information that we can actually process, the stimulations, we can only see a very small portion of this electromagnetic spectrum. And so therefore our perceptions in terms of our visual information is extremely limited.